Okay, this is a voice recording for connections. We're going to try to make as many connections as we can, going through looking at our vital connection in our ecosystem with plants, the autotrophs, the ones that are able to turn the reactions of the sun into viable sustenance for the rest of the ecosystem. So let's start talking about the stuff that we all end up having issues with. Photosynthesis, cellular respiration first. So we'll start with photosynthesis, and we'll connect it to cellular respiration. We'll talk about where we can see these things happening in plants, as well as animals, and then we'll continue on into different aspects of the plants and how it can all connect to our entire year here of AP Bio. So let's start photosynthesis. So as on the left you can see here, this is a chloroplast. So this is going to be your organelle that is going to be doing the photosynthesis for you, or for a plant. Now keep in mind that there are photosynthetic organisms on this planet that are not plants. We have cyanobacteria, that do similar types of things that the chloroplast does, but they are single-celled bacteria. So they harvest sunlight just like the chloroplast does in a environment where they're single-celled. They're going to go through, instead of mitosis and meiosis like a plant would, they would be going through a binary fission. So we have this organelle that looks very similar to a cyanobacteria, but it lives inside plant cells. We end up connecting back to the endosymbiosis theory. And that is the pieces that help us with understanding that is this chloroplast and then even this mitochondria. They have their own chloroplast DNA in here that allows it to replicate itself within a plant's general cell. So let's talk about the compartmentalization that goes on inside chloroplast, and then how it connects to the process of photosynthesis. And we'll make some connections back to mitochondria and talk about it in the big perspective of where these things are happening in the plant. So photosynthesis, we're going to take in light energy. We're going to do the light reactions or light-dependent reactions in the thylakoid. So think of the chloroplast as a double membrane organelle. We have this outer membrane that you can see here, and then this inner membrane that looks kind of like stacks of chips, right? Maybe like a poker chip type of thing. You can see it in multiple different perspectives depending on what textbook you're looking at. But these stacks are called grana, G-R-A-N-A. And those stacks of this chloroplast is where we're going to end up seeing our light reactions going on. This is a thylakoid membrane. And in that thylakoid membrane, remember, anytime you think membrane, you should be thinking these little boogers, right? These little phospholipids where we have a phosphate head and fatty acid tail, so we have a polar and a non-polar, making an amphipathic molecule. Right? And that amphipathic molecule has dualness to it, in that it has a polar edge that allows it to be in the aqueous water solution, and then we have this non-polar tail providing a barrier, so it's going to be tail to tail in this thylakoid, and also the outer membrane of this chloroplast, allowing for us to compartmentalize what's going on. Form-fitting function here. And so this thylakoid is going to separate the inside, right? So think of these as hollow, and this outside, this outside area we call the stroma. So the whole purpose of the light reactions is taking this light energy, and we're going to start making things for the light independent, or a lot of times they say it the dark reactions, but that's kind of a bad term because this does happen during the daylight hours. But the light reactions, or the light dependent reactions, are going to make ATP, high energy ATP, and NADPH, P for plant, right? That's the easiest way to remember it. So NADPH, much like NADH that we see over here, and FADH2, are going to be electron shuttles. So think of NADPH as this molecule that can carry very high energy electrons to power our system. So we're going to use this very similar to how we would use ATP when we're doing an active transport system. So the light comes in from the sun, and we'll break down the two different light reactions that we see in photosynthesis, and it's going to make these ATP and these NADPH. And the way that they're going to do it is based on this separation. We'll go into it in a little bit more detail when we make the connection. So these ATP and this NADPH are sent over to the light-independent reactions in the stroma. So we're not using a membrane here. And there's going to be an enzyme in here which is labeled right here, Ribisco. Ribisco is going to use the enzymatic properties that it has in the reactants that are present to go in. So think substrate, products type idea, active site, maybe even some allosteric promoters and inhibitors involved. And CO2 is going to be bound into some molecules that are already in here. We'll talk about this piece. The water is more used for our intensive purposes in the light reaction. But then eventually you're going to get this G3P. G3P is glycerol 3-phosphate, glycerol aldehyde 3-phosphate, sorry. And two of these G3Ps will make us one glucose, which is C6H12O6. So the whole purpose here is to fix in atmospheric CO2 into a glucose high energy product. Notice that the light independent reactions then feed back the low energy ADP and NADPH. So here's a cycle that's going on, right? We have equilibrium and balance in this system because we're recycling and reusing. This system does not work unless we have continual input from the sun. So if that stops, this whole process stops. And we'll talk about how that connects over here to our cellular respiration. Now plants do cellular respiration as well. Reminder that plants have chloroplasts and they have mitochondria, because the plant has different areas that are green and it has areas that aren't green, so it's going to have to use both of these processes. So we're going to talk about glucose being fed in. So remember that all the glucose is going to happen from photosynthesis. So glucose is going to turn to pyruvate. Note that this is in the cytosol. So if you're thinking about an animal cell, this would be the outer air, or the jelly-like fluid that is separated between the cell membrane and the nucleus. So it's just that jelly area. And then we have the mitochondria, much like the chloroplast. It is double membrane. So we have this cristae, this inner membrane, and this outer membrane, all made up of these phospholipid bilayer pieces. So the whole purpose of glycolysis is to make a little bit of ATP. We'll talk about how it makes it substrate level wise, and then it makes some high energy NADH. Now, in order to get to the next step here, oxygen has to be present. So this is our O2 line in order to get in. If it does not, the plant cell can actually ferment as well. So the plant does need some oxygen, but we'll talk, there's plenty of oxygen inside the plant because oxygen tends to be a waste product. So this pyruvate then moves into the matrix, right, which is the very, very inner part of the mitochondria. We're going to crank out the Krebs cycle. In turn, we'll release CO2, and we'll talk about the connections of CO2 and glucose here in a second. And we make all these high energy NADHs and FADH2s, much like we talked about over here. NADPH, we're going to be sending high energy electrons over to run the electron transport chain. Overall goal, right, we do get some ATP out of this Krebs cycle, but we're mostly going to get all of our ATP from oxidative phosphorylation. So let's talk about why a plant would want to be doing cellular respiration in some areas when it could clearly make plenty of ATP just from its photosynthetic processes. So, blank slide here. The whole connection, we're going to talk about glucose, C6H12O6, plus 6 O2s will yield 6 H2Os and 6 CO2s. Now this is the formula for cellular respiration. So we're going to be taking in glucose like we talked about. We're going to be taking in oxygen. Oxygen in turn gets reduced or gains electrons and turns into water. And glucose gets oxidized and turns into CO2. The reverse process is going to happen for photosynthesis. So this is more exergonic, where we're going to get lots of ATP out of this. And when we're talking about photosynthesis, we got to add energy in to go the opposite way. It's not going to take ATP from the surrounding solution. It's going to take in sunlight. So let's talk connection pieces here. There's obviously going to be some shuttles that are going to be As things get oxidized and reduced, those electrons, in order to not waste too much energy, we do it in more of a stepwise fashion. So there's going to be some shuttles like NADH and FADH2 that are going to help us with this process. And then we're going to end up getting all this ATP because we're going to do it in a stepwise fashion. So if this is our cell membrane, specifically the cristae, we're going to have integral proteins, right? We're making connections here, where we could drop off these electrons, like NADH, dropping off the electrons become NAD+, and those electrons will pop between these proteins, allowing for active transport, specifically of H+, creating a proton gradient. So now we have separation of space. These H+, even though they're small, cannot come back through the cellular membrane, so they have to come through special ports. It just so happens that we have a special port that is an enzyme ATP synthase. Those H+ move back through the ATP synthase to make us copious amounts of ATP. Now that's cellular respiration. So a plant, why would it be doing this? Well, if you think about a plant, right, let's just think a tree, and we'll come back to trees a little bit more in a second. But up here in the canopy of the tree, lots of green photosynthetic organs, right? Doing a lot of photosynthesis. They're taking in CO2. They're producing O2. That's fantastic for us, right? And in turn, they're making all this glucose. Fantastic. It's powering itself. But way down here in the bark, or even lower down here in the root system, right? These roots are providing water for this system for photosynthesis, but they're getting no sunlight. So they can't be doing photosynthetic activity underground. So these cells, even though they're plant cells down here, you wouldn't expect to see chloroplasts in these cells. In turn, you see mitochondria. So these glucose molecules are then shuttled down to the roots, specifically in phloem, right, which is like a artery or a vein that you would see in an animal. It's basically an artery vein piece that's in a plant. So that phloem will allow for that sugar to sink down from the source up here in the leaves down to the sink down here in the roots, and the plant roots can do cellular respiration. So we got mitochondria down there at the roots. All right, talking photosynthesis, we're going to reverse it. We're going to take in CO2 from the air. We're going to take in water from the roots, and those water molecules are going to be the vital source of electrons for us. Now they're low energy in this molecule. This is a low energy molecule, but we can energize it with sunlight. That's the whole purpose of the sun. We're actually going to high energy boost those electrons, and then those high energy electrons are going to power the pumps that we see in the thylakoid, allowing us to make lots of ATP that we can send over to the dark reactions. In turn, we get the C6H12O6, the glucose, or if you're looking at an intermediate step, it would be G3P, and we get waste of O2. O2 can be used in other parts of the plant or nearby, but generally up in the canopy here, we have plenty of energy, so we're not going to be using it for glucose breakdown because oxygen is the final electron acceptor if we're talking about this system here of cellular respiration. Oxygen's going to have to be here to grab that low energy electron, and in turn comes back to water. So note the cycle here, right? So the water that gets used by the plants can be provided by cellular respiration, right? The CO2 that we exhale as waste gets used by plants to make glucose, right? The glucose that they make powers the entire ecosystem, but without the sun, we can't make that glucose. Now the coolest part is when you talk about like a seed of a plant, here's a little teeny seed, and we'll see some pictures of seeds later, in order to become this gigantic tree that you cannot lift, all that mass has to come from somewhere. No mass can be created nor destroyed. It simply just changes form. So where does all that mass come from? Bam! CO2. It doesn't come from minerals. It doesn't come from water, because that tree, a log, could be totally dried out, and the majority of that plant then is cellulose, which is a polymer of glucose. So all the mass of plants actually comes from atmospheric gas that gets fixed by our great buddy, the visco, the most abundant enzyme on the planet. So note the cool connections that are there. That's pretty neat stuff. Let's talk about connections as far as pigments go. Um, we had talked earlier, the reason that you see the green that you see in a tree is because that is the wavelength of light that is reflected back to your eye. So it's actually using all the other, if we're going to do a little spectrograph here, all the other wavelengths of light, so Roy, just not G, and then BIV, right? So we've got the energy wavelengths that are lower in energy, and then the energy wavelengths that are higher, but not in the green area. So how does a plant use, because if we're talking about non-cyclical, if this is chlorophyll A, the energy of the electron gets popped up, rolls back down an electron transport chain, and then gets fed back to another chlorophyll molecule, and eventually ends up in an NADPH molecule. Remember, we feed in the water for the low energy electrons, and light has to be harnessed here with chlorophyll A in order for this whole process to work. So why is it then, for this one, this is Acer rubrum, red maples, why do they look green in the summertime, but at fall it turns this beautiful red color? Well, that's because accessory pigments are present in plant organs that are doing photosynthesis, specifically the leaves, and that will help pick up different wavelengths of light. So we'll get all these what are called satellite pigments or antenna pigments. They could pick up different wavelengths of light and pass it along to chlorophyll to allow this whole process to work. Kind of a cool connection. All right, let's start talking cellular division. So we're talking mitosis. Remember, we have PMAT, right? So prophase, metaphase, anaphase, telophase. But that's all a little teeny wedge in the cell's cycle. So the cell cycle here would be M, and this is not drawn to scale by any means, shape, or form. But we'd have G1, S, and G2. And G1 is growth, and S is DNA replication, and G2 a little bit more growth. So if we're talking chromosomes, single chromosome or a single copy of DNA, when we get to the end of S, we'd have these chromosomes that have an identical copy. Remember, that's called a chromatid. We still see that G2, but at the end of mitosis, they're back to this single-sided chromosome. So plants have DNA just like us. They have these chromosomes just like us. When they do cellular division, they're doing it possibly to repair themselves, but a lot of times they're growing. And you think about organisms, in order to be alive, they might need to grow through their environment, and a big part of that is their ability to divide. So this dividing process allows plants to actually move through their environment. If you want any examples of this, just check out a tree or a bush that's growing near a fence, and you can actually see, a lot of times if this were the fence rail, you can actually see the plant has completely grown around that rail and become actually part of the plant, which is a pretty cool phenomenon. I wish I would have grabbed a picture before I started this recording for you. So let's talk about these dividing areas. So there are areas where mitosis is specifically going to happen in a plant. We have them illustrated here at the apical meristem. So actually, plants only gain length at these apical meristems. So there's ones at the tips of the plant, hence the apic. So, and then we have them at the roots, right? So we have the tips of the shoots and at the tips of the roots. So the plant can actually only gain height at the tips. So if you had a tree, and don't do this, and you nailed a nail into the side of the tree, right now, graduation, and you came back after you finished your PhD, you got your doctorate, you come back and look at that tree, and that nail is going to be exactly same spot. But what's going to happen is, since that plant is aging, it's going to add mitosis at the vascular cambium too. So actually, you kind of see this in the red. It will add new xylem and new phloem, allowing the plant to get thicker. So there's rings on the outside that are going to add new phloem, or ones that add food, and then there's going to be new xylem that adds it to the inside. That provides the ring. So from year to year, it'll add new vascular cambium, and every year it'll actually gain shoot pieces as well. Beautiful time to see the new apex growth of a plant is to check out spring. Right? We can look at some of that stuff later. All right, so growth triggers. Let's talk about some of the different types of growth. So this first one is showing you a fantastic picture of gravitropism. Gravitropism is exactly what it sounds like, growing towards gravity. So we have these things called stratoliths a lot of times in the root ends, and it allows the plant to sense gravity and grow its roots towards it. So regardless of the position of the seed, the roots will always grow downward. Another one is phototropism, which is growth towards the light. The cool part of this is how this all plays out. So in gravitropism, if this were the shoot, the stratoliths would all, based on gravity, come down here to the bottom of the plant because gravity is down here. Right? So we'll say the center of the Earth's down here. And in turn, it'll start mitotically dividing on this side of the plant. So the root will start growing downward. But in phototropism, as you can kind of see here, if the light is here, we actually get not more divisions on the side where you think the light, you actually get more divisions and elongation of the plant on the dark side, right? This is all caused by hormones. Plants have hormones too, like auxin. Auxin is a very common hormone that allows for cellular division. So this is just showing you a lab setup of how they tested for that auxin piece. All right, reproduction, last piece here. I'll talk about how plants are all connected in their meiosis pieces. So plants have two parts of their life. They have a gametophyte and a sporophyte. So you can see sporophyte is when it's got multiple cells in the diploid sense, and then gametophyte is when it's in the haploid sense. So here we have just a regular angiosperm that has a flower. We have the sporophyte, which would be the entire, let's say this is a lily, which you'd see growing above the ground, and below the ground is all sporophytes. All the cells are diploid at this spot. But there's a male part, right, which is the anther, and a female part. And in those different areas, we're going to make the gametes, all from some mitosis and meiosis segments. So here you can see the sporophyte diploid is going to go through meiosis to make the eggs that are going to get fertilized. And same thing will happen up here in the male parts. So here's the ovary, by the way, which ends up turning into the fruit. And then we have the anther that's going to make the pollen grain. And that pollen grain is going to be all haploid pieces, but it's multiple cells in there. So we consider that the gametophyte. But this pollen grain is going to provide what would be like the sperm, and that sperm will stick to the stigma of the female part. It'll drill a pollen tube and dispense two sperm. When it dispenses two sperm, one of the sperm is going to fertilize the main egg, and the other one fertilizes what's called the endosperm. Endosperm is similar to a yolk for a mammal, and that endosperm is going to provide food for the newly fertilized egg that is now going to be the zygote that you can see here, and it will develop into the embryo. Right, so we have the food supply, that endosperm, and that's going to allow that embryo to grow above ground. So we have these what are called baby leaves. They're not the true leaves yet. We call them cotyledons. That'll help start providing photosynthesis. So the seed generally needs to do some mitotic divisions, cellular respiration here, mitochondria, to allow it to get above ground, use these mitotic divisions before it can actually start producing food for itself. So just make those connections if you can. I'll try to make a couple more connecting plants and animals in the future, but we're already at 24 minutes, so thanks for listening.